This animation is about the structure of a gene. If you have a look at the image here, you will see that a gene is made up of DNA in which we can group into exons, which are the expressed regions of the gene, and introns, which I like to think of as the in-between exon regions of the gene. The introns are not expressed in the final protein sequence made from this gene. So if we start at the 5' direction of the gene, the first thing that you will come across is what's called the promoter region. The promoter is the region of the gene which has the sequence which allows transcription factors to come and bind and initiate transcription of the gene. The transcription initiation site, or the start point of transcription, is at the very start of exon 1. The very first section of exon 1 will therefore not be translated because you will always need a section of the gene to allow for the translation machinery to bind to then translate that transcript into protein. So within often the first exon, but not always, you will find a translation start site where you'll find the codon ATG. It's the first codon within the coding sequence. At the other end of the gene, you will find a corresponding translation stop site where the translation stop codon exists. And then there is a section that would be within your RNA but not coded for in your protein, which is your 3' untranslated region. So that's coded for within the DNA, usually within the last exon. And then at the end of that is the transcription termination site. So that is the point at which you no longer make mRNA from your DNA sequence. So all in all, the gene structure is exons and introns, exons being the expressed regions, introns not making it into the final sequence of the protein, so they don't code for the final sequence of the protein. And within those exon regions, you have transcription start and transcription end sites, and then within that region you will have a translation start site and a translation end site.